Hi, I'm Dr. Vicki Websterwood, and on behalf of myself and my co-authors, Drs. Jeffrey Gill, Peter Thomas, and Hillel Cheel, it is my pleasure to present our work on real-time prediction of multifunctionality in Aplysia californica feeding using a Boolean model framework. Our goal is to be able to predict and control multifunctional behavior in real time. To achieve this goal, a tractable animal model and a computationally efficient model framework are needed. Our solution was to develop a hybrid Boolean continuous model of feeding behavior in the tractable animal model Aplysia californica, shown here. Aplysia demonstrates three primary multifunctional behaviors during feeding: biting, which is an unsuccessful attempt to grasp food; swallowing, which is a successful grasp and ingestion of food; and rejection, in which inedible food is pushed out of the feeding apparatus. During each behavior, we can assess the key neurons and muscles involved on an individual basis. For example, in swallowing, 4 phases are observed, beginning with pinching, in which muscles are activated to hold the food so that it is not pushed out of the feeding apparatus. The next phase is protraction, in which the protractor muscle pushes the grasper forwards. In phase 3, the grasper is closed to hold on to the food. And finally, the retractor muscles are activated to ingest the food. The combined activity of these muscles generates force on a strip of seaweed during swallowing. Each boxed burst of activity shown here can then be approximated as Boolean bursts that are either off or 0, representing quiescence, or on or 1, representing firing. Based on our team's own prior work and the existing literature, we then build a model of the neural circuitry in layers. First, the motor neuron layer is developed, composed of key neurons identified from our recordings. Next, an interneuron layer is added. This layer contains buccal interneurons along with their connections to the motor neuron layer. Finally, the cerebral interneuron layer is built. Key cerebral buccal interneurons known to be involved in the multifunctional behaviors were included in this layer. External sensory cues are used to drive behavioral switching, and the motor neuron layer is connected to a simple biomechanical model that provides sensory feedback to the motor neuron and buccal interneuron layers. A simplified continuous 1D biomechanical model is used to represent the quasi-static motions of the grasper and head during feeding. During swallowing, a simulated seaweed is present and fixed to a rigid force transducer. A force threshold allows us to specify a load at which the seaweed will break. Here, an animation shows a schematic representation of the phases of motion during swallowing, starting at rest, followed by protraction with the grasper open, grasper closure, retraction, and a return to rest. Because the seaweed is fixed to a rigid force transducer, retraction of the grasper during ingestion results in forward translation at the head. The model qualitatively reproduces the 3 key multifunctional behaviors. When there is no stimuli in the grasper, but there is positive chemical and mechanical stimuli at the lips, the model reproduces biting. Biting is characterized by strong protraction of the grasper followed by weaker retraction with the grasper closed. If instead, mechanical stimuli are present at the lips and grasper, and chemical stimuli is present at the lips, the model reproduces swallowing. Swallowing is characterized by strong retraction as force is applied to the object. Additionally, the model can reproduce rejection, when mechanical stimuli is present at the grasper and lips, but chemical stimuli is absent, as might be seen if the animal is attempting to ingest a plastic tube. Rejection is characterized by strong protraction as force is applied to the object in order to push inedible food out of the jaws. Behavioral switching can also be simulated in response to changes in external sensory cues, as shown here, for a transition from bite-like to swallow-like behavior. The model can also reproduce multifunctionality within a given behavior by simulating the effect of varying seaweed strength. When we simulate weak seaweed, the grasper moves through a protraction-retraction cycle. As the grasper begins to retract, the force on the seaweed increases until at a given threshold it breaks. On the other hand, when stronger seaweed is simulated, as the grasper retracts, the force on the seaweed increases until the grasper opens. As a consequence, when we compare the retraction duration between the weak and strong seaweed simulations, the cycle is longer when mechanical load is higher, as seen in our animal model. In summary, our model framework is capable of qualitatively producing multifunctional feeding behaviors. It is easily extensible and runs faster than real time. This work was supported by the NSF and the NIH. Thank you.